continuing from our last lecture where we left. Okay, today I'm going to start with the bacteria Salmonella. Okay, Salmonella, it's an gram-negative rod-shaped bacteria. Okay, and if the motility is present, they are usually motile by means of peritrichous flagella. And Salmonella, basically, as you know, it's an pathogen. Okay, there are also other species of Salmonella which are not pathogen, of which one of the most common pathogen is Salmonella typhi. Okay, is Salmonella typhi. Okay, uh, now what are the specialties of Salmonella? This is a photograph of an Salmonella typhi here. Okay, and this is the colony. These are the colonies of the Salmonella typhi. Now this one is a Shigella and this one is a Salmonella. Okay, if you want to grow Salmonella in a lab, okay, you can take a sample of the sewage and streak it on the Salmonella Shigella agar. Okay, and stick it on the Salmonella Shigella agar, and these both agars are basically XLD agars, which are also used for the differentiation of the Salmonella and Shigella. You can see clearly the difference between Shigella and Salmonella colonies, okay, and the Salmonella's colonies. Okay, so these are the various different types of bacteria. Okay, various different types of bacteria, different types of agar. Okay, this one is a Salmonella, black colored colonies. Okay, yes, if you grow Salmonella in the nutrient agar medium, you will get colorless colonies, okay, which are very tiny. Okay, and as compared to E. coli, you can differentiate Salmonella using SS agar also, Salmonella Shigella agar also. Okay, and this one is an red Serratia marcescens bacteria, okay, which have pigmentation of red, and this one is an Escherichia bacteria, Escherichia coli, okay, where you clearly see pigmentation of pink color. Okay, and Streptococcus enteritidis on hecton agar, okay, Salmonella, okay, again on a different type of agar, and this one is an Haemophilus influenzae on the chocolate agar, Now moving forward with the Serratia, as you all know, one more point regarding Salmonella. As you all know, they cause disease typhoid. Okay, but there are more than two species which are involved in the disease of typhoid. More than two diseases which are involved in the typhoid. Okay, of which Salmonella typhi is one of the most common pathogen, and there are also other species which is Salmonella paratyphi A and Salmonella paratyphi B also, which is also an pathogen, and they can be isolated from the sewage. Okay, as well as if they are present in the human beings. Okay, if they are present in human beings, especially in the condition of a disease, you can isolate them from the stool sample also. And moving forward with the Serratia, they are pigmented bacteria which are red in color, again are gram negative cocobacilli or short rod shaped. They are facultative anaerobic, and the chlorate is reduced anaerobically by Serratia, and nitrate reductase is also used. Okay, and one important note to be made: the anaerobic growth does not occur with the chlorate, and have optimum pH of about 5 to 9, and almost all strains can grow with a temperature range of 10 to 36 degrees Celsius. and there are no special requirements for the Serratia strains. Okay, growth requirements. Now, which are the products basically produced by the Serratia species? Acetone, okay, glucose will be fermented, okay, and maltose, mannitol, trehalose are also fermented, and D-alanine, alanine, 4-aminobutyrate, caprylate, citrate, L-fucose, D-glucosamine, kynurate, alpurine, putrescine, tyrosine are also utilized as they are sole carbon sources. Now, dulcitol, tagatose are neither fermented nor utilized as sole carbon sources. Butyrate and 5-aminovalerate are utilized as the sole carbon sources. Okay, are not utilized as a sole carbon sources. So there are a variety of enzymatic activity which is being found in the case of Serratia. And enzymes catalase reaction is positive. Okay, if you want to study catalase reaction of a bacteria, you can use 3% hydrogen peroxide. Okay, and habitat, they occur in natural environments such as soil, water, plant surfaces, etc., and are opportunistic human pathogens. Okay, they generally do not cause disease. Okay, generally if they are involved, they may be involved in the case of secondary infections only. This one is a colony of a Serratia. Okay, where as I told you they are red colored colonies. Okay, they are red colored colonies. Now moving further to the Shigella, they are gram negative, straight rods and are non-motile. Okay, and they are pathogens, and tropism, they are chemorganotroph. Okay, they generally can grow in the absence or in the presence of oxygen also. They ferment sugars, okay, but they will not be able to produce gas, and enzymes, they are catalase positive, there are some exceptions, and oxidase negative, as I told you, they are found in man. Okay, and some of them are pathogens. Okay, and may cause a bacillary dysentery Okay, uh, students who would be opting for the third year, uh, in which they take up microbiology. Okay, you will study in your semester 6 in more detail regarding the Shigella and which disease does it cause, etc., etc. Okay, now this one is a stain of in Shigella. Okay, and these both photographs are of SS agar, okay, in which one contains Salmonella and the another contains Shigella. Okay, if you want to isolate Shigella, you can isolate them using the sewage sample also. Okay, where in the case of Salmonella you will have black colored colonies. In this you have some brown like or sometimes colorless colonies in the Salmonella Shigella agar. Now moving forward with the next bacteria, which is an Aeromonas. Cells are basically straight, rod shaped with rounded ends to the cocoid, and they have about 0.3 to 1 micrometer in diameter. They occur in singly or in pairs or short chains. Generally motile by a single polar flagellum. Okay, and peritracheous flagella may be formed on the solid media in the case of young cultures. And no specific resting stages are known, and specialized structures, they do not have any specialized structures. They are chemorganotrophic and using a variety of sugars organic acids as their carbon sources. Organic acids, sugars as their carbon sources, and they are facultative anaerobes, and have both type of respiratory and fermentative type of metabolism. They have respiratory and fermentative type of metabolism, and their optimum temperature is around 22 to 28 degrees Celsius, but are not able to grow at a temperature range of 35 degrees Celsius. They are not able to grow at 35 degrees Celsius. Carbohydrate can be broken down into acid or gas. Okay, and they also convert nitrate into nitrite. They also convert nitrate into nitrite, and enzymes, they are oxidase positive and as well as catalase positive. They are mostly found in the fresh water and also in the sewage, and they are pathogenic, okay, but to the frogs and fish. They are pathogenic to the frogs and fish. Okay, this one is a colony on blood agar and maconchi agar. Okay, colony of Aeromonas. This one is a blood. This one is an blood agar. This one is an maconchi agar. Okay, in blood agar, normally sheep blood is used. Normally sheep blood is used during the agar, and in the chocolate agar also blood is used. Okay, but the main difference lies in its sterility. Okay, as you all know, the blood of a human being or sheep is usually sterile. Okay, it will be not sterile only in the case of disease, only in the case of disease. Okay, why blood agar should be used? Because it contains some specific components which may enhance the growth of a particular bacterium, This is how blood agar works. Okay, and chocolate agar also contains blood, but why it is of brown color, okay, which you will study in your practicals during your practicals of your semester 3, whoever the faculties who are going to teach you. Okay, and this one is a staining of an Aeromonas. This figure is a. Now moving to the next bacteria, which is an Vibrio bacteria, Okay, as the name itself says, they are vibrioid in shape. Okay, and sometimes they are straight. Okay, and motility in liquid media by means of monotrichous or multitrichous polar flagella, and they are enclosed in a sheath continuous with the outer membrane cell wall. Okay, and on the solid media they may synthesize numerous lateral flagella with a wavelength shorter than that of a sheathed polar flagellum. Okay, in special structures, normally evolutions present in all cultures, and they do not form endospores or microcyst. Okay, this one is a photograph of a Vibrio. Okay, now as I told you, you may also know that Vibrio cholerae is also present in contaminated waters. Okay, and Vibrio cholerae is also responsible for the cholera disease in human beings. Similarly, there are other species of Vibrio, okay, which can also emit bioluminescence. Okay, which can also emit bioluminescence. One such example is basically is of Vibrio fischeri. One such example is of Vibrio fischeri. Okay. Now next group, Haemophilus. Okay, Haemophilus. They are minute to medium-sized coccobacilli or rods, and generally have less than 1 micrometer in width and variable in length, and sometimes forms thread or filaments showing the marked pleomorphism. pleomorphism. They are generally non-motile and are chemoorganotropes. Oxygen requirement, they are aerobic or facultative anaerobic and have optimum temperature of about 35 to 37 degrees Celsius. These Haemophilus species are basically pathogens. Okay. Now, as they are, I told you they are pathogens, and especially they are obligate parasites. Obligate parasites. Okay. It means they will only grow in the cells of the host. Okay, in the cells of the host. Similarly, we can see the same requirement for their growth. Okay, they require various growth factors which are found in the blood, particularly X factor, which is also known as protoporphyrin or protoheme factor, and or V factor, which is an nicotinamide adenine or NAD phosphate. Okay, even after specific growth factors have been provided, growth is normally found in the case of complex media. They are able to reduce nitrites. Okay, and all species attack carbohydrates fermentatively, yielding acetic acid, lactic acid and succinic acids are the end products in the case of glucose broth, are the end products in the glucose broth, and the enzymes oxidize and catalyze reactions may vary according to the type of strains. Among the strains, some may have oxidase positive, some may have catalase positive, some may have both the enzymes positive. Okay, in the habitat, the mucous membranes of main and variety of animal species. Okay, and lifestyle, they are obligate parasites. Okay, this one is a photograph of an Haemophilus as seen here. Okay, and this one here is in colonies of Haemophilus on the blood agar, colonies of Haemophilus on the blood agar medium. Okay. Now moving further with the next bacterium, a colored bacterium, the name itself says Chromobacterium. Okay, name itself says, which is a Chromobacterium, often with bad or bipolar staining and contains liquid inclusions. Okay, normally round in shape. Okay, round in shape and round at ends, as well as some of them are also rod shaped. Motile by means of a single polar flagellum and usually 1 to 4 subpolar and lateral flagella. 1 to 4 subpolar or lateral flagella. And tropism, they are chemoorganotropes and oxygen requirement. They are facultative anaerobes having a fermentative attack on the carbohydrates. And pH, they have a pH optima of around 7 to 8, but they are not able to grow at a pH below 5. Some of them are pathogens also, okay, which causes infections in humans, humans as well as mammals, and temperature requirement around 25 degrees Celsius, but different species have their own different optima, maximum minimum temperature requirements also. Okay, growth factors are generally not required and may utilize citrate as carbon source and ammonia as nitrogen sources, but growth is affected. Okay, but the growth is affected. Okay, they will grow slowly. Acid, but no gas is produced from glucose and certain other carbohydrates, and lactate is oxidized to the carbon dioxide, and nitrate and usually nitrite are reduced often with a visible gas production, and ammonia is formed from the peptone. Ammonia is formed from the peptone. Our enzymes, they are usually oxidase positive, okay, catalase positive and phosphatase positive. Okay, and unique features, they are resistant to benzypenicillin, okay, and also sensitive and are sensitive to tetracycline. Why do we need to go for sensitivity test of the antibiotics? It helps us to identify bacteria also. Okay, it helps us to for the identification of bacteria, as some are maybe resistant to some of the other antibiotics, some may be resistant to other bacterial antibiotics, or some may be also sensitive also to a wide variety of antibiotics. So it may differ. In habitat, they are found in soil, or in water also. Okay. Now this one is an photograph of a colony of a Chromobacterium violacum. Okay, this one is a photograph of the Chromobacterium violacum. Okay. Now moving for today's group. Okay, moving forward with the today's group, which are gram-negative, anaerobic, straight, cowed, and helical bacteria. Okay, which includes I have shortlisted some of them. First one is Acetivibrio, next one is bacterioids, next one is Wolinella, and the next one is Pelobacter. And the next one is the Pelobacter, in which Wolinella, they are generally gram-negative and have a morphology which is helical, cowed, straight, unbranched cells, and about 0.5 to 1 micrometer in diameter, and 2 to 6 micrometer in length with rounded or tapered ends, and rapid and have a darting motility is by means of single polar flagellum, and they do not produce endospores, and on the solid surface medium their colonies are pale yellow, opaque to grey translucent with the convex pitting and with a spreading variance. Okay. Originally, Wolinella basically was described as anaerobic, but in fact a hydrogen and format requiring microaerophile that is capable of respiring with the oxygen has also been isolated. Okay. There you can also use hydrogen and format as the electron donors and are also used as energy sources, and sometimes fumarate and nitrate are used as a electron acceptors. Fumarate and nitrates are used as a electron acceptors. Products: the format is oxidized to carbon dioxide while the fumarate is reduced to the succinate, and the carbohydrates are not fermented and do not support growth, and the hydrogen sulfide is produced, and the catalase negative. Okay, they are catalase negative and are oxidase positive, and they contain cytochromes. Okay, cytochrome is one of the most important compound, okay, which is found in the electron transport chain. Okay, you will study more about the cytochromes if you select microbiology, okay, in your semester 5 and 6. And the habitat, the strains have been isolated from bovine rumen and also from human gingival sulcus and also from dental root infections. Okay. These are the photographs of the Wolinella. Now, next bacteria which is an Acetivibrio, okay, as we are discussing gram negative ones, they are slightly cowed to straight to slightly cowed or occurring in single pairs, and some chains of cells up to about 40 micrometer long. Okay, chains are not the size of the cells, and the motile by means of a single polar flagellum, which are basically located about 1/3 of the distance from the pole to pole by multiple flagella, which are arranged linearly and often faciled and emanating from the concave side of the cells in the both cases. And specialized structures, they are non-spore formers. They are chemoorganotropes, may or may not metabolize peptone. Okay, and acetic acid is one of the sole or major organic acid end product of the carbohydrate fermentation. Okay, due to which its name is Acetivibrio. Okay, it's an obligatory anaerobic organism. With an optimum temperature of 35 to 37 degrees Celsius, and the products, basically formic acid is produced by some of the strains, and propionic or butyric succinic or lactic acids are generally not formed. Yes, these propionic and acids and butyric acids are found in various other fermentations also. Okay, and the other products of carbohydrate fermentations are hydrogen, carbon dioxide, and even ethanol, and they are found in the sewage, sludge, and also in the pig feces Okay, now moving forward with the next group, which will be the last group of the today, which are basically sulfur bacteria, okay, of which the first one is Desulfurella acetovorans, Desulfobacterium, Desulfobulbus, Desulfococcus, Desulfomicrobium, and Desulfomonas pigra. Okay, and this is a very diverse group of bacteria. Okay, some of them are also involved in the photosynthesis. Starting with the first bacteria, which is a Desulfobacterium. Okay, and the bacteria related with the photosynthesis we will study in discuss in the next lectures. Gram staining, they are gram negative and with a shape from oval to rod shape, have single polar flagellum or are non-motile and tropism, they are chemo-organotrophs and can use formate, acetate, propionate, butyrate, higher fatty acids, lactate, pyruvate, alcohols, benzoate, and similar various aromatic compounds as their carbon sources. And they can also use these compounds in the case of anaerobic respirations. Okay, they can use these compounds instead of oxygen for their respiration, and these compounds are completely oxidized to the carbon dioxide. Sulfides and other oxidized sulfur compounds serve as an terminal electron acceptors and are reduced to the hydrogen sulfides. In the absence of external electron acceptor, growth occurs by the fermentation of lactate or pyruvate to acetate and propionate, and some of the species which can utilize hydrogen are generally autotrophic, which can use carbon dioxide, okay, as their carbon sources. They are found in marine, brackish or freshwater sediments, okay, marine but where there is less amount of oxygen, okay, as they are strictly anaerobic, both have fermentative and respiratory metabolism, and respiratory with by the use of sulfates, and utilize proteins, peptides, carbohydrates facultatively by sulfur respiration or by fermentations, and have a pH optimum ranging from 6.6 to 7.6, and their optimum temperature is around 20 to 30 degrees Celsius, and occasionally they may grow up to temperature of 35 degrees Celsius also. And nutrition requirements are very diverse of this Desulfobacterium. Okay, many species use hydrogen or formate and higher monocarboxylic acids up to C16 or lactate and ethanol as carbon donors. Even the several species use aromatic compounds such as phenyl substituted organic acids or phenolic compounds or N-heterocyclic compounds. Okay, so some of the unique features basically are sulfates, as we are discussing sulfur bacteria, and possibly sulfite or thiosulfate are reduced to the hydrogen sulfide. So at the end of the respiration, where sulfur compounds will be used, the end product of the sulfur will be hydrogen sulfide. End product will be hydrogen sulfide. And this one is a photograph of a Desulfobacterium. This one is a photograph of a Desulfobacterium. Okay. Now next one is a Desulfomicrobium. They stain gram negative and morphology from ovoid to the rod shape. Motile by means of a single flagellum. They are chemo-organotrophs and are strictly anaerobic and have a both respiratory and fermentative type of metabolism. Okay, and similar to the Desulfomicrobium, hydrogen gas or lactate or malate is oxidized as an electron donor also, and have a pH range from 6.6 to 7.5. Okay, 6.6 to 7.5, and a temperature range of about 25 to 40 degrees Celsius. Okay, 25 to 40 degrees Celsius. Requirements: they can grow in simple media, but growth stimulated can be stimulated by using yeast extract. Organic substances are incompletely oxidized to the acetate, and ethanol is not used, and desulfoviridine is absent. Okay, which is which is a product in desulfomicrobium. Sulfate, sulfide, thiosulfate are reduced to the hydrogen sulfide, and sulfur may also be reduced. Okay, and similar to other anaerobic bacteria, they are found in anoxic freshwater and marine sediments, and also in case of oil filled waters. Okay, now I am